Imagine a robot, but not just any robot. This one is different. It possesses something remarkable, a brain organoid, a tiny cluster of living human neurons grown in a lab. This is not science fiction, but a glimpse into the rapidly approaching future of robotics, powered by the fusion of biology and technology. This new frontier is made possible by a groundbreaking innovation, brain-on-chip technology. This technology allows scientists to cultivate and integrate living brain tissue with electronic circuits, creating a new breed of biohybrid robots. The implications are profound, promising to revolutionize fields ranging from medicine to artificial intelligence. But this fusion of biology and technology also raises profound ethical questions. As we stand on the cusp of this new era, it is imperative that we proceed with caution and engage in thoughtful dialogue about the potential consequences of creating machines with living human tissue. The future is unfolding before us, and it is our responsibility to shape it wisely. At the heart of this revolution lie brain organoids, often referred to as mini-brains. These tiny structures are grown from human stem cells, capable of transforming into any cell type. Scientists coax these stem cells to develop into neurons, the brain's fundamental units. They develop techniques to encourage cells to form three-dimensional structures, mimicking the human brain. This is where brain-on-chip technology comes into play. These chips house and nurture brain organoids, providing a controlled environment. By placing electrodes, scientists can detect electrical signals, bridging biology and technology. The development of three-dimensional brain organoids represents a significant leap forward in our ability to study the human brain. Unlike traditional two-dimensional cell cultures which grow in a flat layer, three-dimensional organoids more realistically mimic the complex structure and organization of the brain. This transition to three dimensions is facilitated by a variety of techniques, often involving the use of scaffolding materials that provide structural support for the developing organoid. These scaffolds act as a framework, guiding the growth of the neurons and encouraging them to form connections in a three-dimensional space. The result is a more sophisticated and functional organoid, capable of exhibiting more complex behaviors and responses. While still a far cry from the complexity of a fully developed human brain, these three-dimensional organoids offer an unprecedented opportunity to study brain development, disease, and the effects of drugs and toxins in a more realistic and controlled environment. Integrating living neurons with electronic circuits presents a unique set of challenges. Biological systems and artificial systems operate on vastly different principles. Neurons communicate through a complex interplay of chemical and electrical signals, while electronic circuits rely on the flow of electrons. Bridging this gap requires innovative approaches to facilitate communication between these two disparate worlds. One of the key challenges lies in creating a biocompatible interface that allows neurons to grow and function in close proximity to electronic components. These interfaces must be carefully engineered to ensure the health and viability of the neurons, while also enabling efficient signal transduction. Another challenge is the development of sophisticated signal processing algorithms that can interpret the complex electrical signals generated by neurons. These algorithms must be able to distinguish between different types of neural activity and translate them into meaningful commands for electronic devices. This is where the fields of neuroscience and computer science converge, requiring a deep understanding of both biological and artificial systems. Section 5. A symbiosis of biology and technology, grafting organoids onto chips. The integration of brain organoids with electronic circuits reaches its pinnacle with the grafting of these living tissues onto specialized electrode chips. This delicate procedure involves carefully placing a brain organoid onto the chip's surface, which is embedded with an array of microscopic electrodes. These electrodes act as a two-way communication channel, allowing researchers to stimulate the organoid and record neuronal activity. The organoid forms connections with the electrodes, integrating into the electronic circuitry. This fusion creates a hybrid system processing information uniquely. The recorded signals provide insights into neuron communication and responses. Section 6. The learning machine training AI with living neurons. The integration of brain organoids with AI systems holds immense promise for the development of more sophisticated and adaptable machines. Unlike traditional AI algorithms, which are programmed with specific rules and instructions, organoid-based AI systems have the potential to learn and adapt on their own, much like a biological brain. This learning process is facilitated by machine learning algorithms, which are designed to identify patterns and make predictions based on large data sets. In the context of brain-on-chip technology, these algorithms can be used to analyze the electrical activity of the organoid and identify patterns associated with specific inputs or behaviors. For example, by exposing the organoid to a series of visual stimuli and recording its neural responses, researchers can train a machine learning algorithm to recognize those stimuli based solely on the organoid's activity. This approach, known as neural computing, leverages the inherent learning capabilities of biological neural networks to create more intelligent and adaptable AI systems. Section 7. Ethical Considerations: Navigating Uncharted Territory. The development of brain-on-chip technology and the integration of living human neurons into machines raise profound ethical considerations that we, as a society, must carefully consider. These ethical implications are far-reaching, touching upon our understanding of consciousness, sentience, and the very definition of life itself. One of the most pressing concerns is the potential for sentience or consciousness to emerge in these hybrid systems. While current brain organoids are far too rudimentary to possess anything resembling consciousness, the rapid pace of advancements in this field necessitates a proactive discussion about the ethical implications of creating systems with the potential for such capabilities. Another ethical consideration is the source of the stem cells used to create these brain organoids. Currently, most research relies on donated stem cells, but the potential for using stem cells derived from other sources, such as embryos, raises additional ethical concerns. The use of such materials must be subject to rigorous ethical guidelines and regulations to prevent abuse and ensure responsible innovation. Section 8. A Future of Possibilities: Repairing and Enhancing the Brain. Beyond robotics, brain-on-chip technology holds immense promise for revolutionizing our understanding and treatment of neurological disorders. The ability to grow and manipulate living human neurons in a controlled environment offers unprecedented opportunities for studying the mechanisms of brain diseases and developing new therapies. One area of active research is the use of brain organoids to model neurological diseases such as Alzheimer's and Parkinson's. By introducing specific genetic mutations or environmental factors into the organoids, researchers can recreate aspects of these diseases in a dish, allowing them to study their progression and test potential treatments in a more controlled and ethical setting than is possible with animal models or human subjects. Another promising avenue is the development of brain-computer interfaces, or BCIs, that could be used to restore lost function in patients with paralysis or other neurological impairments. By implanting electrode arrays into the brain, researchers hope to create a direct link between the brain and external devices, allowing patients to control prosthetic limbs or communicate using their thoughts. Section 9. The Hybrid Mind Merging Human and Artificial Intelligence. The convergence of brain organoids and AI raises the tantalizing possibility of creating hybrid intelligences, systems that seamlessly blend the computational power of silicon with the learning and adaptability of biological neurons. Such hybrid systems could potentially surpass the capabilities of either humans or machines alone, leading to breakthroughs in fields ranging from scientific discovery to artistic expression. However, the development of hybrid intelligences also raises profound ethical and philosophical questions. Section 10. The Ethical Imperative: A Call for Dialogue and Responsibility. As we stand on the cusp of this new era of biohybrid robotics and augmented intelligence, it is more important than ever to engage in a thoughtful and inclusive dialogue about the ethical implications of these technologies. The decisions we make today will have far-reaching consequences for the future of humanity, shaping our relationship with technology and redefining what it means to be human. We must proceed with caution, guided by a deep respect for human dignity and a commitment to responsible innovation. This requires open and transparent communication between scientists, ethicists, policymakers, and the public. we must ensure that the development and deployment of these technologies are guided by ethical principles that prioritize human well-being and prevent unintended consequences. Furthermore, we must invest in education and outreach efforts to ensure that the public is informed about the potential benefits and risks of these technologies. An informed citizenry is essential for making sound decisions about the future of these powerful tools. A new era of discovery and ethical exploration. The fusion of brain organoids and AI chips marks the dawn of a new era in robotics and artificial intelligence. We must proceed with excitement and caution, driven by knowledge and respect for ethical implications. The potential rewards are immeasurable. By harnessing these technologies responsibly, we can unlock new frontiers in medicine and AI. Let us shape it wisely.